Okay, so folks, we have a lot to cover today, and I want to make sure that you get through the final section on CRISPR-Cas9, especially in relation to in vivo work. That's something that I believe will be important for you, not just on the final exam, but also as a technique. It's going to become a more and more standardized technique, and I'll show you my rationale for why I think that it will be just as useful and just as powerful as optogenetics is for most labs currently. So we'll go back and we'll take a look at genome remodeling, and then I'll sort of segue into things that you don't have to necessarily worry so much about, and I'll talk to you a little bit about how you might want to get into finding research. If you haven't done research yet, as an undergrad student, I very highly recommend that you get into research as early as possible. It gives you a different perspective and a better understanding of the types of things that I talk about here in lecture. And then I'll talk a little bit about what I hope you will decide to do down the road. That self-serving lecture that I talked to you about on Tuesday, it's actually self-serving because I want you to sort of link that with what I'm going to be telling you here today. So I'm going to go back and tell you a little bit more about CRISPR. I'll give you a little bit of a review from Tuesday's lecture, and then we'll talk about how CRISPR can actually be used in vivo, and then I'll go through some of the experimental details. I can't go through all of them. It's actually a short paper, but the supplemental figures that are on that paper actually are longer than the paper itself, and so I can't take you through everything, but I'm going to take you through some of the experimental details because they are really important. We'll start wrapping up the course, and I'll remind you about a couple of upcoming assignments and things that you need to make sure that you are taking care of, and then we'll go through a general overview of the final exam, and then I'll talk to you a little bit about what's next for you as a student. Okay, so we had left with one of these types of diagrams in the last sort of lecture, and we had talked about some of the basic considerations that you had to take into account when you are designing some of these guide RNAs. This red portion here is now something that you don't actually have to worry about cloning. It comes ready and prepared for you. And all you have to do is design this blue portion, and you have to worry about things like GC content, the overall length. The longer it is, the more specificity there is, but at the same time, there are also potentials for other types of interactions that I mentioned in the last lecture. There was one paper that came out toward the end of last year where this was also used in vivo. It's a much simpler system. I know it sounds somewhat disingenuous to say that, but it's a simpler system in the sense that you can directly inject this particular virus into the liver, and because of the way that the circulation into the liver through the hepatic and portal system works, that it's easy to target this large organ. So there actually has been one study in vivo using CRISPR, which is outlined for you here. And I went through like the brief basics of all of this in the last lecture, and I'll go through this construct again in today's lecture. Right now, more and more individuals are kind of looking beyond sort of these traditional methods of delivery, whether it's an adeno-associated virus or whether it's a lentivirus. All of these different types of technologies have limitations. There are limitations in terms of size. There are limitations in terms of the types of molecules that you can clone in. Some are RNA dependent, some are DNA dependent. And so what people are looking at now is sort of the next generation, which is beyond this, which is nanoparticle delivery. And nanoparticle delivery and cationic lipid delivery is probably going to be introduced in the next 3 to 4 years. So you'll hear more about that wherever you end up going. So I told you that this is one of the first papers, and that's actually a tour de force. If you ever want to look at behavior, cell biology, genetics, if you wanted to look at how you do next generation sequencing, this is the type of paper that you would want to do for a journal club. And this is one of the first papers that actually use CRISPR in an intact animal system, and it's only one of 5 papers that have actually been used in the nervous system, and this is the most complex one out of all of the ones that have been published so far. So I'll take you through that in a little bit. And this Nature Biotechnology paper is something that you might want to go back and read. You don't have to. I'm going to take you through the things that I want you to learn from this particular paper. I can't go through all the details, to be quite honest, because I need a full lecture to be able to do that, and I don't want to give you a full lecture on your last day of class. This Cas9 actually exists in a number of different bacteria. So one of the different species of bacteria is this Streptococcus, and this Streptococcus Cas9 has been the dominant type of Cas9 that's been used up until this point in time. And I'll go into that in a few moments. And this SP, or Streptococcus pyogenes Cas9, has previously been used in vivo, in vitro, to cause those frame shifting insertion deletion events, and then the DNA repairs itself through homologous recombination. So we'll be looking at this sort of knockdown effect again today. And again, most of the studies up until this point in time have either used a really simple tissue model, or they've been using replicating cells, or they've been using cell lines. The effects in neurons, which are not replicating and they're not the type that normally divide, has been much more challenging as a result. When I say that neurons don't divide and they're post-mitotic, what does that mean for all of you? Yes? Well, they are already differentiated, right? So what state of differentiation are they? Sorry? Yep, they're all in G0. They stay in G0 or G naught. And in G naught, these post-mitotic neurons have always been a real challenge for neurobiologists. Because they are sitting in a weird place in the cell cycle, it's hard to really transfect neurons. We don't get genes into them easily because they don't tend to divide. So we can't really manipulate them as easily as we can for cell lines or cell culture models or for cancer cell lines. And it's been a problem for a while. However, this paper and others have actually taken that into account. So again, this is actually the first model system that used a hydrodynamic injection into the tail vein of these mice, allowed this particular vector using Cas9, it's still the SP Cas9, and the delivery of the guide RNA. This is actually a simple model system, and I'll have to take you through this in a little bit more detail in the upcoming slides. But this is all in one vector, and you have to remember that. So when they express Cas9, they're also expressing the guide RNA. There's no doubt that if you have Cas9, that you also have this guide RNA in the same cell. It's all in one adeno-associated virus. It's all packaged together, and when it targets the liver, it will target each individual cell with Cas9 and the guide RNA to knock down the particular gene of interest, okay? This is going to be a little bit more complicated in the model that I'm going to show you in the brain. So if you have any questions, today is the day to ask, because it is something that's really important for you to understand. So in order for this particular paper to address some of the issues and some of the technical challenges and hurdles that were facing them, they decided to use this SP-Cas9 system. So it's been used before. It was used in that previous hydrodynamic injection paper that I just showed you, or the cartoon from that paper. And for using this in the nervous system, can you think of a reason why we might want to use AAV? So again, thinking back as well as forward to your final exam. Why might you want to use AAV? What are some of the advantages that AAV confers? There's technical ones, and then there are some more sort of research-based ones, like bigger picture ones. Have you seen other papers that use AAV and inject into the brain? So you know that it works, right? And you know that the efficiency of AAV works really well in the brain. So that's one reason for using it. And the other reason for using it is that when we're creating these guide RNAs, it's easier to work in the AAV system than, for example, the lentiviral system to work backwards to generate Cas9. So although these AAV vectors have some technical limitations, and one of it is size. Like one thing that if you go back and you look at that lecture slide that had all of the different types of vectors, whether it's herpes simplex or whether it's AAV or whether it's lentivirus, some of them change in terms of length of expression or speed of expression, and some also are different in terms of the size of the construct that you can physically put into the adenovirus. Do you remember that slide? It's an important slide for you to make sure that you remember for your final exam as well. Because of the size of SP-Cas9, it's been a real limitation up until this point. It takes up most of that viral packaging. And even though this guide RNA is fairly small, it makes it somewhat challenging to actually deliver this all in one vector, unlike that hepatocyte vector that I showed you before. So they're going to use, in this particular paper, they're actually going to inject in two different vectors or two different adenoviruses, one that contains the Cas9 and then the other one that contains the guide RNA, okay? So normally we would have them together. In this particular paper, because of size limitations associated with AAV, they can't do that. So they put two different things together in two different AAVs, and then they inject those. Victor, you have a question? It's being expressed as in the AAV. Then it's going to get into the cell. The cell is going to transcribe and translate it, and then it will be expressed there. It's not a protein. So the latest research that's being done right now is to actually deliver fully functional proteins into the brain using nanoparticle delivery. But that's many years off. You won't be probably seeing many papers like that for the next few years. Yep. Yep, I'm gonna show you. I'm gonna show you that on the next diagram. Any other questions before we go on? So one of the big issues on this, and it's a really challenging issue, for those of you who have done any kind of transfection studies, when you're trying to take two different plasmids or two different constructs and introduce them into the same cell, how do you know that both of them are going to get into the same cell? And that's one of the challenges that they had to actually address in this particular paper. So Victor's question was related to this. So how do we get this Cas9 in there? So this is one of the constructs that is injected into the dentate gyrus. And this particular construct, and some of the details are found below, this construct is being driven by a MECP2 promoter, a partial promoter. The longer the promoter is, the less we can actually package into this construct. So we have to minimize the promoter to allow for expression driven by that promoter and make it specific. But if we have the full promoter, we actually can't put in all of the SP Cas9. Here is the Cas9 enzyme that we were looking at. It's also being expressed only wherever you get expression of MECP2. And again, there are some nuclear localization signals around it, and then there are these internal tandem repeats. We won't go into the molecular biology behind why we need that, although you can probably figure out we need to target this to the nucleus, which is why we have these NLS signals. That's construct number one. We inject this first, or we can inject this, and then we will also inject at the same time this other guide AAV or SP guide AAV. This is under a different promoter, so it's not the MECP2. This is under the human synapsin 1 promoter, so again, targeting neurons. It should be neuron specific. Here they have GFP as well as the Cache9, cache, and the guide RNA. So wherever you see GFP, you're going to be sure that you have also delivered this guide RNA. Okay, so again, it's a two construct delivery system, and both of these SP9, Cas9, and SP guide vectors are used to deliver both elements of the guide RNA and the Cas9 separately, but you hope that they get into the same cell. All right, is that okay for everyone? Okay, so and again, the vector that encodes that sequence for the guide RNA has this GFP cache, and as long as you're getting expression of GFP, you are most likely getting the expression of the guide RNA. Yep.
How do they know that the Cas9 is in the same cell as where the GFP is?
Okay, how would you do that? I'm going to take you through it, but let's think about that a little bit. So you know you're going to get GFP, and wherever you get GFP, you're going to get guide RNA. How would you go back and check to see whether or not you have Cas9? There are a couple of ways. Yep. Sure, you could see a functional effect, I guess. That'd be kind of expensive. You want to just do a really quick assay. How else could you do it? In theory, is if you had GFP and you had an antibody to Cas9. So remember, it's not something that's you're going to be expressing in large quantities, but they did something actually kind of neat here. And I didn't mention this. I didn't want to make this into a really big thing here. They added what's known as an epitope tag. Normally, we don't have in our cells, we don't have this molecule known as hemagglutinin. Okay, and HA stands for hemagglutinin. It's a very small sequence. It's only 8 amino acids long, which means that it's only about 24 base pairs long, which we can tag this cell. So wherever we have HA, we know we have Cas. We normally have no HA at all in our normal cells. And so this epitope tag we could stain for. And then when we have HA signal along with GFP, then we would know that we have expression of both. And it's a quick, dirty way to do it. Does that make sense to everyone? And this small little epitope tag here is more cost effective than generating antibodies to Cas9. Okay, so this cache domain also helps to direct that GFP fusion construct into the outer nuclear envelope. So again, you're getting the proper expression in the right place of the guide RNA and the Cas9 as well. So again, there are very, very short neuron-specific promoters, both MECP2 and synapsin-1. Both of these are neuron-specific promoters. And I'll go back and review, because you've already had this with Dr. Eubanks, I'll review MECP2 with you very briefly. But this is also considered to be a neuron-specific promoter. We truncate these promoters as small as we can because the size is a limitation in these AAV vectors. And again, we have this truncated version of the MECP2 promoter. We take it down from about 1 kilobase to about 235 base pairs, which makes it much smaller, a quarter of the size. And we put in the smallest possible polyadenylation signal as possible for SPA. And again, that detail, you don't have to worry about numbers or anything else, that it's just important to minimize for packaging. So surprisingly, yesterday, the same group, Feng Zhang, actually came out with another paper in Nature. I don't think he publishes in anything lower than Nature. And he actually has done an amazing piece of work. So he knows that there are limitations with the Streptococcus. So he's actually looking at other bacteria, Staph aureus, and he is looking at all of the different types of Cas9 systems there, and he's actually able to shrink this even more. So in theory, this is becoming a more and more efficient process. And again, you have to think about this. This started with one publication in January of 2013. In 2015, now, the rate of change of this whole technology, this is already the 4th generation technology after 2 years. And again, it's worrisome enough, and you don't have to worry about the details on this paper, it's worrisome enough that this technology is growing faster than any other molecular biological technique that's ever been encountered in the history of mankind, that the scientists in the European Union have actually suggested that we stop all experiments using CRISPR-Cas9 because we don't know what the consequences of this might be. And so that was also big news earlier on this year as well. So this is the power of the technique. So again, he published a paper at the end of 2014 that just got accepted or was fully published earlier in January 2015, and now he has another follow-up paper in March related to an even more efficient form of this CRISPR-Cas9. So this gets into exactly the same question that was asked earlier on. So you have to make sure that you actually have the delivery of both systems to the same cell. Like, how do you make sure that you do so? And it's not 100% accurate. So when you look at the overall efficacy of the dual delivery system, 2 different viral vectors delivering it to the same sort of neuronal systems, one of the things that they started off with, which is actually very common in science, instead of starting the injections directly in mouse, in a mouse brain, they decided to do this in a primary mouse cortical culture. And they did this in cell culture first, in other words, before doing this in a brain. Can you think of a reason why you might want to do that? Simple reasons. Yes?
There's like less compounds.
Sure. Well, it's not a cell line, it's a primary culture. So it's a little bit more like the native brain. So there are cell lines which are sort of immortalized and you can keep growing them over and over again. And then there are other ones that you remove and you grow up from the embryonic stage all the way up, and you can only keep them around for finite periods of time. There's a simpler reason. Where do you think it's easier to count cells? In a cell culture system like this where they're lying down flat, or in a brain? It's probably a lot easier to count the efficacy in a flat culture, which is what they decided to do. So again, they went in, they tagged with HA, this epitope tag. They have an antibody to HA, and since HA is infused in frame with Cas9, wherever you get HA signaling, you have Cas9 being expressed. And then if you go back and see where the guide RNA is being expressed with this GFP cache, which is also on a different vector, and then you merge the signals and you look at all of the different cells that are here, you'll see that the efficacy is somewhere around 75%. I'm not really sure because I looked at the data that they had in their publication. I'm not sure if that's supposed to be 75% as shown here, but in the text, they mentioned that it's about 75% that they saw with the co-expression. So it's not 100%, but when you deliver two different viral particles together and they get into neurons, there's about 75% co-transduction or co-expression of these two different vectors. The other thing that's really important is when you are doing this, does that Cas9, which is normally not overexpressed in these different systems and not in mammalian systems, does it affect the morphology of the neurons? And no changes in morphology, no changes in the health or the physiology of these neurons were encountered. So again, it looked like a safe system. Not great, not 100% efficiency, but 75% is certainly considered to be great. So one of the things that they wanted to do is, can they start going in, just like your colleagues mentioned, and can they actually see, we've got this in, it's in at about 75% efficiency. Can we now go in and see whether or not it actually has an effect on the genome? So we've got this thing in there. Does it really work, in other words? So again, they were targeting this X chromosomal link gene, the MECP2, that methyl CPG binding protein. Remember that it is really important. This particular protein is really important in Rett syndrome. And MECP2, as you may or may not recall, although for the final exam you should, it's ubiquitously expressed in neurons throughout the entire brain, cortex, hippocampus, dentate gyrus, everywhere. And if you have a deficiency in this, this is associated with the morphological and electrophysiological phenotypes that Dr. Eubanks mentioned in passing to you. All of these electrophysiological changes that we see when you have a deficiency in MECP2 are thought to contribute to the phenotype that we observe in Rett syndrome. So these guide RNAs even though we're using the MECP2 promoter to get it into neurons, there's nothing else associated with MECP2. They also wanted to go in and use guide RNAs to knock down the function of this MECP2 gene. Okay, does that make sense for everyone? So they want to see if they can knock down the expression using this new CRISPR technology, can they actually affect at a protein level and then at a behavioral level the effect of MECP2? If MECP2 is gone, in other words, can they make something look like they have Rett syndrome? Okay. So again, you don't have to worry about this. I wanted to just draw this to your attention. You don't have to memorize all of this. This is more to take you back to the very first slide that we had today, which is when you're generating those guide RNAs, there are some limitations, and those are limitations that you should be comfortable with the CRISPR and Cas9. I told you it's not a perfect system. You need these PAM sites on the opposite strand for CRISPR to work. If you don't have these and you don't have this guanine in the 5' position, it doesn't work and you can't target it. Luckily for MECP2, you can actually target a number of these sites within the gene. And when you target a number of these sites, some of them work better than others, which is what I'm going to try to show you down here. So these are all of the different types of guide RNAs that were used. to different targets. And you can see that when you start getting these different types of bands, these are sort of the insertion deletion effects that you can start to pick up. So you have almost no effect here. There's only about a 5% occurrence of what's known as an indel, an insertion deletion. So it stops the production of that gene expression. There's nothing detectable in the control, nothing detectable using another guide RNA, nothing detectable over here as well for this guide RNA, but other ones have a lot more of an effect, like 23% detection of different indels. So when they go back and they start looking at the expression of MECP2 as a protein, now on a Western blot, one of the things that they see very clearly is that they can knock down the expression of the protein if they allow CRISPR and Cas9 to work in these different cultures, okay? And you can see very clearly that when they have this MECP2 guide RNA, the one that worked at the highest efficiency, not the ones that don't work, but the highest efficiency one, and they also have the presence of the Cas9 to allow the guide RNA to work, they are almost able to completely eliminate the MECP2. And again, it shows very strikingly that in this system that they're able to offset the expression of MECP2. And I think just for the time being, we'll skip through that one particular slide. So they also wanted, again, to see that is at a biochemical level. It's always important to show that if this works, it should knock down the expression of a protein. They showed that. They showed that it causes insertion deletion events through what's known as, do you guys, have you all taken like a biochemistry course? Do you know what a nuclease protection assay is? Sort of, yes. Some of you are shaking no. You can use what's known as a nuclease protection assay to see the overall in the overall integrity, for example, of the nucleus and some of the genes that are being expressed. And that's one way to do it. But this is actually the best biochemical evidence that at a protein level, this is being knocked down. So on a different level, morphologically, when you add this, the most effective guide RNA, and you allow the Cas9 enzyme to go to work, what sorts of phenotypic changes do you see? So remember, in Rets, with Dr. Eubanks, we saw lots of phenotypic changes at the level of the dendrites, at the level of the spines, at the level of learning as well. And when we start going in and taking a look, that overall there is a big change in the number of dendritic arborization. So it's not as complex. The other thing that you can see up here is that when you have the MECP2 guide RNA, not the control RNA, this is just a control that they have to be able to do. And when they have the MECP2 guide RNA along with the Cas9, you see a dramatic reduction in the number of spines here on the bottom panel. And again, the spine density per unit length is also significantly reduced in following the knockdown of MECP2 as well. So again, it shows that we have an alteration in the dendritic morphology, spine numbers, the different axonal crossings, as well as the number of dendritic ends, the overall complexity of the dendritic tree, just as Dr. Eubanks was mentioning to you. Yeah.
On the previous slide, so is it that the target is no longer the PAM sequence and it's a sequence inside the MECP2 gene? Is that?
No, within the MECP2, it's something that you have to have endogenously in the gene that you are targeting. You need these different PAM sites. And all of them exist in the on the guide RNAs. Like they're all being used to target these PAM sites.
So there's multiple PAM sites in the MECP2.
So again, the gene is fairly big. And when you're looking at that gene site, can you find any of these PAM sites that contain these double guanine residues, and then whatever other residue that follows after it? And if you can, this is one of the rules that you have to be able to follow. And when you find that in your sequence, then you would then take a look, can we target this particular sequence? Yes, we can. Then you would generate a guide RNA to it. No, it does not have to be at the end. It can be anywhere inside the gene. Yep. Okay. So they did all of this. This was all still done in culture, but they weren't able to actually make any kind of claims to doing things in vivo. So all of this is in primary culture. All of this is in neurons. So they're kind of like real neurons, but they're not like the neurons that you would find in your brain. But they still see a pretty dramatic effect. They see a reduction in dendritic arborization. They see changes in spine numbers. They see lots of effects at the protein level. However, they don't really know what happens in vivo, and that's really what they want to be doing. They want to, as was quoted in their article, they wanted to directly perturb specific genes within intact biological contexts. And part of it is I just love the way that Fang Zheng writes. Like, he has a really distinctive writing style when he's writing things up in his publications. Most of the colleagues that I would have been writing with would have just said, we want to see if it would work in an in vivo system, right? But he has this really cool way of saying an in vivo system as an intact biological context. So in this intact biological context, one of the things that he did was he injected this, the double construct, the two different AAV constructs into the dentate gyrus. And he wanted to know, when he goes back and he stains the dentate gyrus in these intact animals and he removes the brain and then he goes back and counts for MECP2, what is there a difference? And indeed, he saw that when he had the specific guide RNA plus Cas9, that there was a decrease to about 70% of the dentate gyrus in the animals that were injected were indeed reduced. And again, the total protein levels, not in culture, but in the intact animal, was also reduced by about 60% within the dentate gyrus. Okay, so again, stunning results showing that in an intact animal system, that he can actually do exactly the same thing, that the efficiency of this system can actually be used in vivo in this intact context. And I'll show you some of the results that he had from his slides. For those of you who have a that on your computer screens, you also get like a really black blob right around here. Yes, and around here as well. Interesting. Weird. Okay, so again, if we go in, this is what normal MECP2 staining. So this is, again, a neuron-specific protein. MECP2 is found enriched here within the dentate gyrus. If you go back and stain these individual nuclei with the DAPI stain, which stains all nuclei, and you go and take a look, this is what the normal pattern would look like. This is what the pattern of MECP2 looks like after you have injected the guide RNA plus the Cas9. You can see a dramatic reduction. It's not completely gone, but there is a dramatic reduction in MECP2. The neurons are still there, by the way. It's just that MECP2 has been decreased. So the number of DAPI cells on this panel on the left and this panel on the right are approximately the same, but the level of staining for MECP2 is dramatically decreased. So it's not a loss of cells. He hasn't killed these cells. What he's done is he's driven down the expression of MECP2. And again, he does other experiments to show that it's MECP2 specific, that it's not knocking down other things. There are no off-target effects. But again, I won't have time to do and go through all of that. So again, this is at the Western blot level. So he isolates the dentate gyrus, and when he takes out the dentate gyrus, and then he runs the dentate gyrus with a control injection series and with the MECP2 injection series, he sees that 60% decrease in the overall expression of MECP2 in the dentate gyrus. And again, the number of cells is also dramatically reduced. It's reduced down to about 30% of what it was previously. And again, it shows very, very specifically that he is able to have a biochemical effect on an intact system. Is that okay for everyone? Yes. The number of MECP2 positive cells. Yeah, not the number of cells. No cells are dying by this injection. Any other questions? It was actually one of the things that's in his supplementary figures. So I'm assuming that one of his reviewers asked him exactly that question. So are you sure that you're not killing cells, that you're actually reducing the expression? Victor, you have a question? Yes. Yep. Yes. Yes. Yeah. That's actually, there are 2 papers now that actually show that you can do rescue effects using CRISPR-Cas9 as well. So they're not in vivo yet, and they're kind of being used in a different system in the cardiac system. But yes, absolutely. Do you have a question? In theory, so again, one of the reasons why about 100 to 150 scientists in the European Union asked for this to be stopped is that with next generation sequencing and whole genome sequencing becoming more common, it's theoretically possible to go in, sequence your entire genome, decide that, for example, we should completely eliminate cystic fibrosis, and we should take out that defective gene and then add back in a good copy of the gene, and we can do this at the level of a zygote, and we can then have someone that will never burden the healthcare system, but they're worried about the technology right now. So yes, in theory, that's exactly what scientists are proposing to be able to do. Yeah. Are the results immediate? So they're typically waiting about 1 week to about 2 weeks to actually do some of these Western blot studies, and these functional studies, which I haven't talked to you about yet, are exactly the same. Yeah. So it's not an immediate effect. It takes time. Yeah. Any other questions? So all of this is still very much biochemical, whether it's looking at the number of stained cells or whether it's looking at total protein levels. It's not in vitro anymore. It's not in cell culture. It's now in the intact animal. It's been delivered intact. They've allowed it to be expressed intact, and then they go back and they see that in the intact animal, it is indeed reduced. So does this, if you knock down MECP2, does it have any behavioral consequences associated with it? Since MECP2 is associated with learning as well as memory, one of the things that they decided to do was to use a contextual fear model system. Okay, and when they did so, they could actually show that when you are training these different animals for freezing behavior after learning that a foot shock is coming associated with a tone in this particular region of a box, that these animals don't freeze as often. They don't really learn as well as they should be learning. So with this knockdown in protein, and the knockdown in the number of cells that are expressing MECP2 and at a protein level, functionally, they're not able to learn this contextual fear conditioning response as well. They don't freeze when they hear a tone in a particular region in that fear conditioning box. Is that clear for everyone? And again, it's only in that particular box that they've learned that there's a tone coming. That's where they're going to get a foot shock, that they get this freezing behavior. If you're wondering about the white bars, the overall freezing, it's not a problem with mobility or anything else. If you move them out of that box into a different box, into an altered context, which are the white bars, they behave just like the control animals do. No freezing or very little freezing behavior, as you'd expect. They freely roam around and explore. So again, all of this is summarized for you here. And again, this has this role in learning, and they decided to use a contextual fear conditioning model that I just outlined for you. And this the dorsal region of the dentate gyrus is one of the structures that's important for this type of learning to occur. So you can imagine why they set out to do this. And when they do so, it's exactly the results that I reported back to you, that it only occurs in a specific context when they have learned that or where they can't learn that association between a foot shock in one part of the box. Okay, and again, these MECP2 mice, other than that, other than this learning deficit that you would expect, have absolutely everything identical to what you would normally assume that mouse would have. They don't have any altered anxiety. They perform normally in an open field test. They have normal novel object recognition. They do well on an elevated plus maze. It's only this one learning paradigm that is associated with the dorsal dentate gyrus, and it's a specific region, and it's a specific behavioral task that is affected. Nothing else is affected. Is that okay for everyone? Yes. Yep, they only did there.
So how can we be sure that if this was, because people with Rett syndrome have other
Yep.
full body effects, so how can you be sure that if it was?
They're not saying that it's a perfect model for Rets. It could be, it might be, but they're showing that it doesn't have any effects that they would not expect to see. So they expect to see effects in the dentate gyrus, but they don't expect to see other things related to innate learning or anything else. Okay. Oops. Okay, so the last thing I'm going to mention to you, and I'm not going to go through this in a lot of details. I have like one more slide left to this, and it's really a heavy looking slide. It's really not that bad. It goes through identical things that I did on the previous slide. One of the things that I told you that the CRISPR-Cas9 system is really good at is that you don't have to target just one. You can target like multiple genes at the same time. And that's one of the beauties of the particular model system that we're looking at. So this is the same construct that we've already looked at before. However, now, one of the things that they wanted to do was they wanted to use that function which I mentioned to you at the end of the last lecture, which is called multiplexing. So multiplexing allows you to take out multiple genes at the same time. It's unlike RNAi, where you typically take down one at a time. You can take out all of them at exactly the same time. And using this multiplex system, it looks a lot like what we've already examined. And here they're targeting different gene targets. These are these DNA methyltransferases that you, I think, heard a little bit about from Dr. Eubanks. And these different DNA methyltransferases, there are three of them that we have within the mammalian system. We have the DNMT1, MT3A, as well as MT3B. It turns out that we don't have to worry about all three. We only have to worry about two of them, and I'll go through that in some more more detail. But overall, it's the same strategy. It's the same type of construct that we've looked at before, but now instead of just trying to take down MECP2, they're trying to take down all of these three DNA methyltransferases. Can you think of a reason why you might want to do that? Because you're really cool and you're Fong Jiang and you feel like you can, right? That's one reason, I'm sure. Yes. In some ways, I guess that's accurate. But so think about sort of the downstream effects that are associated with MECP2, and these are some of the downstream targets that have been implicated with MECP2. All of these DNA methyltransferases are found at different periods of time. So the DNMT1 and 3A, for example, are only found in the adult brain, which is what they're looking at. The 3B is found in the developing brain. Early on, when they're in the neonatal period, they have the 3B form. All of these are implicated in synaptic plasticity, learning, and memory formation. So these are some of the downstream targets for the MECP2. Is that okay for everyone? So there actually are DNMT3A and MT1 knockout mice. They have altered hippocampal dependent forms of memory. So this gets back to your question. So are other systems affected? Yes, potentially there are. And one of the things that makes this multiplex system really unique, if you're thinking about it, you've heard a lot about different transgenic animals that have compensation, right? You knock out one gene and something else takes its place. By multiplexing, you can wipe out everything in that particular pathway, which is what they're trying to do here. So if there are any compensatory effects where another of the DNA methyltransferases takes over, none of them have the opportunity to do so because you're literally wiping out all of them. And they're really careful about this too. They decided that even though the 3B form is not found in the adult, just in case, we're also going to knock out the 3B at the same time as DNMT1 as well as DNMT3A. So when they started doing that, again, the effects are pretty pronounced. They can show at the level of protein that they get really good knockdown of the 3A through Western blots, all in vivo, as well as DNMT1, not quite as pronounced an effect, but still significant. And again, showing that functionally, and biochemically they can knock down this particular protein or proteins. And again, when they look back at the same behavioral paradigm, because they're still looking in the dentate gyrus, when you knock out DNMT1 and DNMT3A and 3B, they behave just like the MECP2 knockout or knockdown animals. They also have this reduced freezing behavior. And come to look at it, the data looks kind of weird here. Sorry, I just noticed something. I want to just check. Sorry about that. Yeah, the data looks weird, eh? Did you see that? Yeah, the freezing behavior is actually really high, much higher than you would normally expect, but you know, you can't really argue with Fong Zheng. So previously on that other graph, if you go back, I won't do that again. I hadn't noticed this until I started flipping through the slides. The freezing behavior now, it's almost like 100%, right? It's like 80% freezing is a lot for an animal. And it is significantly reduced, but it doesn't look like the type of data they had earlier on. So it's not a perfect model. So in any ways, you can't really argue with them, right? So this shows you that this system, which has so much promise right now, has a number of unique properties. It can knock down and multiplex multiple genes at the same time. They are working on rapidly changing the system so that it becomes easier and easier to use. And to be honest, in about a year and a half, this will be used in some of the undergrad labs beginning in second year. So it's that type of technology. Everyone knows that it's really useful. Everyone knows it's really simple to be able to use, and it teaches you a lot about molecular biology or about functional behavioral biology as well. Do you have any questions on this before we go on? Yeah, me?
I'm not sure I get why they wanted to know both of them. Is it because they wanted to make sure they don't compensate?
Yes. Yeah. So for example, if you knock down um, one form or one isoform of a calcineurin, for example, you might have the other calcineurin kind of compensating for it, and you never know if you're not seeing an effect because of that. So if you knock down all three, then you're not going to get that compensation. Any other questions before we go on? No? Actually, I think now is a pretty good time to take a break. So if we take a break a little bit early, I'm going to try to finish off. I'll tell you a little bit about your final exam, and I'll take you through some research and other things, and then we'll wrap things up and we'll take our own class picture. Okay, I'm going to take you through a couple of things really quickly. One is to try to get you in the mindset of getting ready for the final exam. So I've already been asked a question about this. I will be making it very clear what you should know for the final exam on Blackboard as well. Some of you who have at least started organizing your notes, as you know, there's a lot more material on the final than there is on the midterm. The midterm really had about 4 lectures. It was kind of all contained within learning and memory. This one is sort of spread out all over the place. Therefore, it's intrinsically more challenging. You have to know a lot more. There's more material, etc. I did not set out to make the final exam any more challenging. My job is not to make the average lower or anything else. It's really just to see if you know some of the more challenging concepts that we've had here. It's the same format as I mentioned at the very bottom, 20 multiple choice for short answer questions with parts. So the short answer questions, you get 1 page to write things out. 2 of those short answer questions are related to applying some of the techniques that we've been discussing over the last little while. Only 1 of the questions is related to just being able to give me back some details. So that was like the midterm. So the midterm was really, tell me what you remember about this phenomenon, or tell me what you remember about this part of the nervous system. The final exam is a little less like that. It's more about application. There are many ways to answer the question, as long as it's scientifically sound. You'll do fine on that. You only get 2 hours, so those of you who have been coming in and been telling me that you were a little short on time for the last midterm, something to be a little bit aware of. And again, it's in the format of design and experiment to test like neurogenesis, to test Rett syndrome, to test some aspect of what you've learned, or go back and look at Parkinson's disease or other types of things. Before I go on to the next slide, do you have any questions? Yes.
For like the design and experiment type
Yep.
question, will you be getting any sort of
Yes, and I think that's on the next slide. Not the sample question, but like how I'm going to get you ready for that. There was an earlier question about this not being non-cumulative. So none of the details of the material that you had before, I'm hoping you haven't forgotten it, but none of that's going to be tested. So we just had things like the elevated plus maze, the novel object recognition. I'm hoping that kind of rings a bell, but I'm not going to be asking you to outline it all for me. Okay? Yes.
For one of the techniques, like if we were to revisit some of the techniques that you've introduced in the pre-midterm section, like can we do that?
Yeah, you can. In your answer, I don't really care, but I'm not asking you to learn it specifically for the multiple choice or anything else. Yep. If you bring things in from another course and it makes sense, I don't mind that as well. It's perfectly fine. Okay, so I want you to start preparing for the final exam. So some of you are looking at it and it's April 29th. It's a long way from now, so I'm not going to get started until maybe April the 24th. Maybe that'll work for you, maybe it won't work for you. But if you're not doing anything else right now, you should be going back over your notes to make sure that all your notes are complete. And that's like the bare basics that you should be doing right now, like over this weekend. Do I have all the notes? Am I missing any recordings? Have I not listened to any of the recordings? When am I going to budget the time to be able to do that? All of the lectures, including the ones from the guest lectures and TA lectures, are going to be testable. I'm actually going to be posting next week when you have I think you have a study break next week before exams actually start. I'm going to be putting the exam breakdown for you. So like how many questions to expect from, for example, Nadia's lecture. Is it going to be like one short answer? Is it going to be 5 multiple choice? I'll give you the exact breakdown per lecture, okay? And that'll all be available for you, I think Tuesday of next week. So again, the best way to help yourself out is to start getting all of your notes ready and to at least see which ones you're going to have to spend a little bit more time on. I can guarantee you 3 days is not going to be enough time for you to do well. So if you're hoping to get an A, many of you have an A, and again, this year where all bets are off, I can have whatever class average I want. So this is your year to see if we can actually go for an A or A+ average in the course. Let's see if we can do it. I will also, not next week because I'm going to be meeting with students throughout the next week, but I will start having office hours starting the week after. You can always drop in. It'll be an open office hour. And then the other thing, in preparation for the types of questions that you should expect, one of the things that I'll be posting, similar to the midterm, is a participation question. I will actually be evaluating you on this question. It's not just doing it. You have to have a solid answer. You get 48 hours to think about your answer and then write it out. Then I'll be posting all of them anonymously. You don't have to comment if you don't want. At this point, everyone's busy, so I don't want that to be onerous for you. But they will give you a good indication as to the types of experimental design questions that you'll get on your final exam. Is that okay for you? What else do you guys want to do? I'm worried about the long month gap that you guys have.
I just have a question.
Yep.
Participation question.
Yep.
This weekend, will we expect
You don't have to finish it this weekend. I'm going to be posting the questions and then you get 48 hours to finish it off. Sorry? I'm going to be posting the questions sometime on Sunday-ish, maybe Sunday evening, and then you get 48 hours after to submit your answer. Again, it's one question, so yep, yep. Can it be earlier? Friday? Okay, when I'm okay with it. When would you like it?
tomorrow?
Like next weekend, because like I know for me, like right now, it's like I've done some of this, but I've done more like anatomy and the other one. And
How about this? Let's keep it simple. How about we post all of the questions sometime on Friday? And if you can do them early, then do them early. And if you can't do them early, do them before next Monday. Not this coming Monday, but next Monday. Is that all right? Okay, so that was easy, right? Okay, what else? Yep, do you have a question at the back? Yep. Yes. I think we do. I haven't gotten all of the YouTube links up there yet, but I will be spending some time updating everything. Okay. I think I have both audio and do I have not just audio? I have audio only for the guest lectures, but I have like different videos that I have to post up as well. Yep. Any other questions before I go on? Okay, so I'm going to talk to you a little bit about research. Some of you are really interested in it. You're in your third year, so I wanted to talk to you a little bit about it. How many of you are interested in doing research? How many of you are actually doing research now? Okay, so some of you are and some of you aren't. I can tell you that when I went through undergrad here, I did not do any undergrad research whatsoever. And when I got into grad school, it was actually a big shock to me. Throughout undergrad, I was not a great student. I was not even a good student. I was not even an average student. I was kind of a below average student throughout most of my undergrad. I started getting a little bit better in my third and fourth year. I can tell you that getting into research was an accident. Like I graduated and I had no idea that I had no idea. Yes, the remnants of a great and mighty empire. Um, the 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 I had no idea that I was going to go into research. I actually was thinking because I'll show you my next picture with my mom. As we were walking away from convocation, my mom asked me, after I finished my undergrad and I had like less than stellar marks, my mom asked me, so what will you do next? And I said, I don't know. And so for the next 3 weeks to a month after I convocated, I actually spent most of my time sending out my resume to TD Bank, Royal Bank, Scotia Bank, and I was going to like my job, my glamorous job was going to be as a bank teller, and that was sort of what I was hoping to do after I graduated. However, luckily at the time it was possible for me even late to get into research. I did get into research, and even though I wasn't a great student in undergrad, I really liked research. I didn't like being in the lab that I was in, to be honest. I hated going into the lab every single day, but I liked being I like doing things in a research lab. Like I really liked experiments. In other words, I'd never thought about doing experiments, and so my master's was actually a little bit better because I have a little bit of a smile there, and I actually kind of enjoyed it. Um, and that was something that was an eye-opening experience for me. This article that came out in the Wall Street Journal yesterday, if you read at the very bottom, sometimes all it takes is just being where you are. You don't have to do anything. People actually come and give you like jobs, or some people will give you like research positions. But most of you will know that you have to apply for these things. And in some cases, you have to apply a lot, like 300 emails. I've actually heard of students sending out tens to maybe almost 100 emails trying to get like one research position. I'm sure you've heard those stories as well, right? And for those of you who said that you wanted to get into research but don't yet know how to do that, I don't want you to be like me, leaving things off to the very last minute, kind of being lost and not really sure how to get into research. I really lucked out in the sense that I got into a research field really, really late, like in toward the middle of August before the next semester started. You wouldn't be able to do that anymore. So sometimes you're really lucky. Sometimes it takes a lot of hard work. But if you read down here at the bottom, our research community is not really different. The industry is actually very connection based. It's actually, once you get into research, it's easy to get your next research position, but it's that first research position that is so difficult for you to get into. And that got me thinking a lot when I started teaching, because one of the things that I really want students to do from a number of different levels is to do research, because you have to figure out, like me, whether you like it or you don't like it. For example, I hated my master's, and I actually had to take a year off after doing my master's because it was very, very traumatic for me. Like no one wanted me to defend. My supervisor and I didn't really get along very well, and it was a real battle to be able to defend my my master's. But then I decided that I liked research enough that I did my PhD, and my PhD was a very different experience, and it was a very enjoyable experience. It was an experience where my supervisor actually encouraged me to ask all sorts of different questions of him. He gave me all sorts of different opportunities, and so my research experiences were very, very different. But again, the only way that I knew that I would like it or dislike it is by actually trying research. And that got me thinking a lot about undergrads, because I know you're just like this slide that you want to do research and you may not have been able to open any doors yet. And how do you open some of those different doors? And when I started teaching, one of the things that my best friend Harold asked me was, so Bill, you're about to go and teach at U of T, so that's great. Congratulations on that. When you go and teach at U of T, would anyone have known that you actually went to the University of Toronto in undergrad? And I had to think about that, and I said, well, I'm not sure what you mean by that. And my transcript is there, my degree is from there. And he said, but if you were ever missing from a class and you never showed up to a class, would anyone in your class actually even know that you were in that class? I said, probably not. And then he said, how are you going to make sure that you don't do that when you're in teaching? And I hadn't really thought of it that way. And I actually started rethinking about what I wanted to do with students. And I do want you as students to be able to make an impact. I also want to be impacting on your lives as well. And I'll get to that in a moment. Part of this is one of my favorite psychologists out of Stanford University is Carol Dweck. And Carol Dweck has a really interesting way of thinking about how to motivate students not to necessarily get A's. She wants students to get A's because they feel motivated to study, not to have easy courses, not to just get by and slack off. For her, it's about trying to motivate people and for them to be able to make an impact on others in the same way. And so I was really influenced a lot by a lot of individuals. And I want to go through a couple of things with you really quickly if in the last sort of 5 minutes. So I've introduced you to a lot of people throughout this course. Some of it is because I need to do that as part of the course material that I need you to know before you go off into fourth year. Some of you will be taking fourth year pathology courses, some of you will be taking physiology courses, and you'll need this kind of background to be able to do well in those courses. And so some of the lectures were designed that way. But if you remember Dr. Eubanks and you remember Sean, I want you to think about this as a generational effect. So Dr. Eubanks was my prof at one point in time, and I never thought of being a prof. And the same way with Sean coming in to teach. He's a teaching assistant now, but I also want to encourage him to go on to be a prof or do other things in life. And I want you, as you're sitting there right now, to be thinking about maybe in 2 years or maybe even next year, maybe I can come in and teach in your course. And I certainly like that to happen. I'd like to make sure that we're helping you develop all the skill sets that I think are necessary for you. In doing so, as I'm hoping that we create an environment where you guys like coming to class and you know each other in class, that as professionals, if you're going into a professional school or other things, or if you're thinking about getting into research or academia, that I'll be able to help you in different types of ways, whether it's on a small scale level individually or on a program level, or maybe one day, and that's my hope, that the impact will be big enough that maybe we can actually change the University of Toronto. It's a big goal, it's a big dream, and it's sort of a 10-year plan that I sort of have some investment. And that is an investment. It's a long-term plan that I want to have. So some of this gets into helping you study for assignments or do well on tests, or to start thinking about mentoring. Some of it is a really practical thing. I don't want you to ever feel, for example, that you should be ashamed or embarrassed or shy about coming in to see me for a reference letter, because you're all looking to do different things. It took me, for example, over 2 weeks to find 2 individuals to write me a reference letter at the last minute. And most of them were saying, yeah, I don't really feel comfortable about that. But I've known all of you for the past semester. And so for me, I'd love to be able to help you wherever you're trying to go next, to write you a reference letter, get you into a research lab, try to help you connect with other profs who might be looking for research students. Can I help you in other aspects of your university life? So maybe you're thinking about taking on some more responsibility. Maybe you have an idea for a project. Maybe you have an idea for a conference. Maybe I can help you with those types of things as well. And again, in fourth year, as you're starting to think about that, there are lots of awards and other things that you should be applying for, and I certainly would like to help in that process as well. And so these are some of the practical goals that I've set for myself, and that means that out of the 80 or so students that I have here, that I'll have to be meeting with you beyond this course to help you out. For most of you, if you're applying after graduation, unlike myself, who kind of left it to the last minute. If you're applying in a proper way, you're gonna have to start thinking about this actually early on in the fall. Your fall semester is important. This semester was really important, and so will the fall. You're gonna have to start thinking about your courses, how that's going to look on wherever you're going to be applying next. I want you to start thinking about which profs you want to start interacting with, and which profs you want to actually be in courses with, which courses that you feel are going to allow you for mentorship, etc., and build relationships with profs. There's so many great profs in sociology, psychology, in sciences, in physiology, etc. It's really easy to find good profs these days rather than worry about running into the wrong kind of prof. I also want you to find courses that are going to be satisfying. Like again, with Carol Dweck, don't find courses that you've heard you can get a good GPA in. Find courses that are going to evaluate you fairly and that are going to help you in your degree. And again, start planning as early as possible for your postgraduate studies. Don't be like me, in other words. Don't let me be a role model for you. Plan a lot earlier than I did. We won't go into specialized or not. That's already too late for most of you. I think you're all either a specialist now or a major. So by the end of this year, you should already know what kind of courses you like. Like I really hate courses where I have to do lots of writing, or I really hate courses that have multiple choice. I really hate courses where I have to write grant proposals, and you'll have to decide for yourself what kind of courses you want to get into. So learn how to study more effectively, especially in your fourth year. It'll be really challenging. If third year was tough, fourth year, everything will be rushing at you at the same time, at the same week, at the end of November and the end of January. And if you, sorry, at the end of April. And again, if you need to come and talk to me, please do. So what kind of courses do I want you as neuroscience students to take? So again, I want you to take courses that are going to help you in graduate school or medical school, where you're going to be in groups. I know that some of you don't like group assignments, right? You hate group assignments. Yes, lots of nodding heads. That's okay. You're going to have to learn to like group assignments. The bottom line is, wherever you go, whether it's in a profession or whether it's in research or academia, you're always going to have to be collaborating with someone. There's always going to be someone who is not doing things the way that you like them to be doing something, and you have to be able to handle that. So again, think about that. And this is my wish list for you. So if you can take some of these courses next year, it'd be great. And again, you can talk to different individuals who've already taken these courses. Some are more challenging. So if you really want to know about drugs and how they work on the brain, and you want to work with one of my former profs that I had, Dr. Mac Burnham, who's an expert in epilepsy, then you take pharmacology PCL 475. It's a tough course because it's a year-long course, and you learn the mechanism of action of like this many drugs if you stack up all the notes. But by the end of it, you're actually going to be the best neuropsychopharmacologist on campus, to be honest. Like I've had students that go through there and they know more than most grad students do. Take a nutrition course like NFS 489 or LMP, or some of the great physiology courses. I love the CSB courses. There's another one, it's the stem cells course of CSB 430, but this is another great one by Dr. Wooden, seminar-based, where you get to do some presentations. The Psych 490 series are fantastic, and please take a research course. If you don't think you can get into a research course, absolutely wrong. I can help you get into one. Lots of options. If you don't want to do HMB, do another one. Do it in Psych or PSL or CSB or Anatomy. Wherever it is, it doesn't matter. Just do a research course. I'm going to ask you not to take too many HMB courses. I want to see you all in fourth year, but if you're loading up on HMB courses, you're not going to be experiencing all of these other great courses like LMP or PSL or pharmacology. So just take enough to fulfill your neuro requirement. And again, challenge yourselves. In fourth year, it's not really about protecting your GPA as much. Your GPA is just going to naturally go up, I can guarantee you. What do you think the averages are? Oh, you guys are all laughing. Well, okay, I don't know universally, but I can tell you like the averages in my fourth year courses are usually around an A-. And I like it around an A- because I think by that time everyone's pretty proficient. I don't have to tell them like how to do different types of assignments. The tests are different and everything else. And it's not about memorization. It's more about understanding and applying things. So I'm hoping that you will in turn be able to make a really big impact at the university. You've made a big impact on me throughout this semester. It's been fantastic getting to know you. I know I'm going to continue to interact with you over the upcoming weeks. And I know some of you are really nervous about the final exam. Don't be. As I said, it's something that is going to be fair. It's going to be something that's based on the lecture material. And if you have any questions, all of the practice tests, assignments, and everything else are designed to help you get ready slowly for the final exam. And with that, I'm just going to thank you for a great semester. And if you can stick around for a picture, we're going to follow Gianna out towards Sid Smith, and we're going to be at the front of Sid Smith, and we're going to line up on the stairs there. I know it's time out of your schedule, but I would, again, really appreciate it, so thank you. Gianna, can they follow you out toward Sid Smith and then at the St. George side? Thanks.